Let's start by asking a fundamental question. You look around at life on Earth, plants, animals, bacteria, fungi, there's enormous diversity, right? But here's what's fascinating. If you analyze a plant tissue, an animal tissue, or even a microbial paste chemically, you find the same basic elements. Carbon, hydrogen, oxygen, nitrogen tungents, the same ones you'd find in a rock or soil sample. Now here's where it gets interesting. When scientists analyze non-living matter, say a piece of Earth's crust, they find similar elements. In absolute terms, there's no big difference. But when you look more closely, when you really compare the proportions, you notice something. The relative abundance of carbon and hydrogen is noticeably higher in living organisms compared to non-living matter. That difference is key to what makes life special. In fact, the most abundant chemical in any living organism is water. But alongside water, there are thousands and thousands of organic compounds. We call these biomolecules. Now, organisms also contain inorganic elements and compounds. So when we analyze living tissue, we're looking at this complex mixture of organic biomolecules and inorganic substances. That's what we're going to explore in this chapter. So how do we actually analyze what's in living tissue? Here's the technique. You take any living tissue, could be a vegetable, could be a piece of liver, and you grind it with trichloroacetic acid in a mortar and pestle. You get a thick slurry, then you strain that through cheesecloth or cotton. And now here's the key part. You end up with two distinct fractions. The part that passes through and dissolves, we call that the filtrate, or more technically, the acid-soluble pool. And scientists have found thousands of organic compounds in this pool. The part that doesn't dissolve, the retentate, that's the acid-insoluble fraction, it contains the really large molecules. And here's an important point. All those carbon compounds we extract from living tissues, we call them biomolecules. Now, we want to know about the inorganic stuff too, so we do a different experiment. You take a small piece of tissue and weigh it, that's the wet weight, includes all the water. Then you dry it completely, all the water evaporates, and what's left is the dry weight. Finally, you burn the tissue completely. All that carbon is oxidized into carbon dioxide and water vapor and goes off into the air. What remains is called the ash. And this ash, it's full of inorganic elements, calcium, magnesium iron, and inorganic compounds like sulfate and phosphate. All right, let's dive into specific biomolecules. We'll start with amino acids, which are absolutely fundamental to life. Here's their structure. Amino acids are organic compounds that have two special functional groups, an amino group on one side and a carboxyl group on the other. And here's the key feature. Both of these groups are attached to the same carbon atom, which we call the alpha carbon. That's why we call them alpha amino acids. Now that alpha carbon has 4 valency positions, 4 bonds it can make. 3 of those are fixed. One is a hydrogen atom, one is the carboxyl group, one is the amino group. The 4th position, that's variable, and yes, that's where we get the diversity. That 4th position has what we call an R group, and the R group can be different in different amino acids. This is important. There are only 20 types of amino acids that are found in proteins, just 20. Not hundreds, not thousands, 20. Some have a hydrogen as the R group, like glycine. Others have a methyl group, like alanine. Some have a hydroxymethyl group, like serine. Each one different based on what that R group is. Now we can classify these amino acids based on their chemical properties. Some are acidic, glutamic acid is a good example. Others are basic acid, like lysine. Many are neutral, pimeidine falls into this category. And then there are special aromatic amino acids, tyrosine, phenylalanine, tryptophan, thymine. These have ring structures in their R groups. Here's a really important property. Both that amino group and that carboxyl group are ionizable. That means they can gain or lose protons depending on the pH of the solution they're in. So the same amino acid can have different structures at different pH levels. This is crucial for how proteins work in cells. At certain pH values, something interesting happens. The amino acid exists in what we call the zwitterionic form. That's when both the amino group and the carboxyl group are ionized at the same time. This form is typically what you find in cells. It's a special balanced state. Let's shift gears now and talk about lipids. Here's the defining characteristic of lipids. They're water insoluble. They don't mix with water. That's one of the things that makes them so useful in cell membranes. Now, the simplest lipid is a fatty acid. A fatty acid has a carboxyl group attached to what we call an R group. That R group can be quite variable. It could be a methyl group or an ethyl group or longer chains of CH2 groups. The whole chain can range from 1 to 19 carbons. Let me give you specific examples. Palmitic acid has 16 carbons total, counting the carboxyl carbon. Arachidonic acid has 20 carbons. These specific numbers matter because they affect the properties of the fat. Now, fatty acids come in two main types based on their bonds. Saturated fatty acids have no double bonds. All the carbons are fully saturated with hydrogen. Unsaturated fatty acids have one or more carbon-carbon double bonds. Those double bonds actually change how the molecule behaves and how it affects your health. Another fundamental lipid component is glycerol. Glycerol is a simple 3-carbon compound with 3 hydroxyl groups attached in the DAS. That's why it's called trihydroxypropane. It's small, but it's crucial because it serves as the backbone for building more complex lipids. Here's where it gets interesting. When fatty acids attach to glycerol, they bond through a process called esterification. You can get one fatty acid attached to a lactant. That's a monoglyceride. 2 fatty acids, diglyceride. 3 fatty acids, that's a triglyceride. These are what we commonly call fats and oils. Now, what's the difference between a fat and an oil? It's actually pretty straightforward as such. It depends on the melting point. Oils have lower melting points. That's why gingerly oil, for example, stays liquid in winter. Fats have higher melting points, so they're more solid at room temperature. The difference usually comes down to the proportion of unsaturated fatty acids. Now let's look at more complex lipids. Some lipids contain phosphorus, a phosphorus atom, thulone, along with a phosphorylated organic compound. These special lipids are called phospholipids. Phospholipids have a crucial role. They're found in cell membranes. They're the molecules that create the lipid bilayer, that double layer of lipid molecules that surrounds and protects every cell. Lecithin is a well-known example of a phospholipid. Interestingly, neural tissues, your agarity, your nerve cells, they contain lipids with even more complex structures. They're not just simple fats or standard phospholipids. They have specialized structures that are important for nerve function. Now, let's talk about nucleic acids, which are the genetic material. The building blocks start with nitrogenous bases alloy with heterocyclic rings containing nitrogen. There are 5 main ones: adenine, guanine, cytosine, uracil, and thymine. You've probably heard these letters before: A, G, C, U, T, neutral. They're the letters of life's genetic code. Now, when one of these bases attaches to a sugar molecule, we get something called a nucleoside. The names change slightly. Adenine becomes adenosine, guanine becomes guanosine, thymine becomes thymidine, uracil becomes uridine, and cytosine becomes cytidine. But here's the crucial part. If you also attach a phosphate group to that sugar, now you have the complete package, a nucleotide. That's what gets linked together to form DNA and RNA. The names again change: adenylic acid, thymidyllic acid, guanylylic acid, uridyllic acid, cytidyllic acid. These nucleotides, and only nucleotides, are what form the chains of DNA and RNA, our genetic material. Let's take a step back and categorize all these biomolecules differently. We call some of them primary metabolites. These are the compounds you find in pretty much all animal tissues. They include amino acids, sugars, fatty acids, nucleotides. They have clear, identifiable functions and known roles in keeping organisms alive. But in plants, fungi, and microbes, there's a whole different set of compounds called secondary metabolites. These are thousands of different molecules that you don't typically find in animal tissues. Think alkaloids, those are compounds found in plants like coca and opium. Flavonoids are pigments. There are essential oils, antibiotics, colored pigments, scents, gums, spices. It's an enormous variety. Here's the interesting part. We don't fully understand why organisms make these secondary metabolites, but we do know they're valuable. Drugs come from secondary metabolites, spices come from them, scents and pigments, even rubber. Some of them have ecological importance. They might protect the plant from predators, for example. Scientists are still discovering new functions for these compounds. Now here's an important organizational principle. The molecules we find in the acid-soluble pool, the ones that dissolve G, they're relatively small. Their molecular weights range from 18 daltons, that's the size of a water molecule, up to around 800 daltons. We call these micromolecules, or sometimes just biomolecules. The molecules in the acid-insoluble fraction fizzle. The ones that don't dissolve because those are dramatically bigger. Their molecular weights are in the range of 10,000 daltons and above. We call these macromolecules or biomacromolecules. They include proteins, nucleic acids, and polysaccharides. There's one exception though, lipids. Their molecular weights don't exceed 800 daltons, so they should be micromolecules, but they end up in the macromolecule fraction anyway. Why? Let me explain. Lipids are actually small molecules. Their molecular weights don't exceed 800 daltons. But here's the thing, in living tissue, lipids don't just float around individually. They organize themselves into structures. Cell membranes, for example, are made of lipids arranged in bilayers. Now, when we grind up tissue and subject it to the acid fractionation, something happens. We're disrupting all those organized structures. Cell membranes get broken into fragments. Those fragments form little spheres called vesicles. And here's the key. These vesicles are not water soluble. Because they're not soluble in the acid solution, they don't dissolve. They stay with the insoluble material. So even though lipids are small because they're organized into membrane structures that form water-insoluble vesicles, they end up in the macromolecular fraction during our separation technique. But this is important to remember. Lipids are not strictly macromolecules. They're in the insoluble fraction because of how they're organized in the tissue, not because they're inherently huge molecules. Let's pull back and see the big picture here. That acid-soluble pool we get from fractionation sponges, it roughly represents the composition of the cytoplasm. It's got all those small molecules, dissolved ions, simple organic compounds, anarchists, basically what you'd find floating around in the fluid inside the cell. The acid-insoluble fraction represents the macromolecules, the large molecules, from the cytoplasm and from the organelles. That's your proteins, your nucleic acids, your polysaccharides, and those lipid membranes. These are the structural components that give the cell its architecture. Together, the acid-soluble pool and the acid-insoluble fraction represent the complete chemical composition of any living tissue. And when you add it all up, when you look at what's most abundant, water wins. Water is by far the most abundant chemical in any living organism. Everything else happens in that aqueous environment. Now let's focus on one of the most important macromolecules, proteins. Proteins are called polypeptides. That means multiple peptides strung together. What's a peptide? It's amino acids linked together. And they're linked by something called a peptide bond. So a protein is essentially a chain, sometimes a very long chain, of amino acids all connected by peptide bonds. Now remember those 20 amino acids we talked about earlier? Proteins are made from those 20 types. You can think about whether a protein is a homopolymer or a heteropolymer. A homopolymer has only one type of monomer, repeating over and over. But proteins aren't like that. Each protein typically uses many different types of amino acids. That makes proteins heteropolymers made from different types of building blocks. The science has been clear for a long time, but the evidence from this past decade is definitive. You know, this period from 2015 to 2025 will be remembered as the decade the consequences of our inaction became undeniable. That temporary breach of 1.5 degrees served as a final, unambiguous warning. Proteins do an enormous variety of jobs in living organisms. Some transport nutrients across cell membranes, they carry things in and out. Some fight infectious organisms, those are antibodies. Some act as hormones signaling molecules that coordinate activity between cells. Some are enzymes, we'll talk much more about those. Some proteins are structural, those they form the framework of tissues. Some are receptors, they receive chemical signals from other cells. There are thousands of different proteins, each with its own specific job. And here's a remarkable fact. The most abundant protein in the entire animal kingdom is collagen. It's the protein that gives strength to your skin, your tendons, your bones. But here's something even more remarkable. If you ask what protein is most abundant on the entire planet, in the entire biosphere, it's called RuBCO. That's ribulose bisphosphate carboxylase oxygenase. It's the enzyme that plants use to fix carbon dioxide during photosynthesis. Because every photosynthetic organism on Earth uses it, and there are so many plants, this one enzyme is incredibly abundant globally. You probably have some of it in your body right now from eating plant material. Let's look at another major class of macromolecules, polysaccharides. These are long chains of sugar molecules. Not just one type of sugar, but different monosaccharides serving as building blocks. These chains are found in the acid-insoluble fraction, which tells you they're big molecules. Imagine a cotton thread that's actually a good analogy for polysaccharides. They're literally threads. But instead of cotton fiber, they're made of sugar units connected end to end. Depending on which sugars are used and how they're arranged, you get different types of polysaccharides with different properties. Let's look at a specific polysaccharide, a cellulose. Cellulose is a perfect example of a homopolymer. It's made of only one type of monosaccharide, glucose. Just glucose, 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 repeated over and over in a long chain. The chains are so long they form a major structural component. Plant cell walls are made of cellulose. That's what gives plant cells their rigid structure. That's why a carrot is crunchy, why plant stems are stiff. Paper, that's made from plant pulp twamp. It's cellulose. Cotton fiber, cellulose. This is an incredibly important structural material. Here's an important point about cellulose's structure. Unlike some polysaccharides we'll see, cellulose forms a linear structure. It doesn't coil up into helixes. Because it doesn't have that helical structure, it can't hold iodine molecules in little pockets. So cellulose doesn't turn blue when you add iodine. That's a key test to distinguish it from other polysaccharides. Plants need to store energy and animals need to store energy. They do this with polysaccharides. Starch is how plants store glucose. Glycogen is how animals store glucose when splik. You have glycogen stored in your liver and muscles right now. There's also inulin, which is a polymer of fructose used by some plants. These are all energy storage molecules. Here's where starch is different from cellulose. Starch forms helical structures. It coils up like a spring. These helices create little pockets or cavities, and this is crucial. Iodine molecules can fit into those pockets and get trapped there. When you add iodine to starch, you get a deep blue color from that starch-iodine complex. That's how we test for starch. Cellulose, on the other hand, we already established it's linear. It doesn't form those helices, so it can't trap iodine. No blue color. This difference in structure, helical versus linear, explains why starch is great for energy storage, while cellulose is great for structure. Same basic building block, glucose. But different arrangements lead to completely different functions. Beyond simple glucose chains, there are more complex polysaccharides with more interesting building blocks. Instead of just plain glucose, they're built from amino sugars with amino groups attached, AVARCs, and other chemically modified sugars. Glucosamine and N-acetyl-galactosamine are examples. These create more intricate, specialized molecules. A famous example is chitin. This is what makes up the exoskeleton of arthropods, insects, crustaceans, spiders. It's a strong, rigid material that provides protection. It's also found in fungal cell walls. Chitin is complex, but interestingly, it's still mostly a homopolymer biohumidity. It's mainly made from one building block, just not plain glucose. It's made from N-acetylglucosamine. We talked earlier about nucleotides and nucleosides in passing. Let's now look at how they're organized into nucleic acids. Nucleic acids are polynucleotides, chains of nucleotides. They're part of the true macromolecular fraction, grouped with polysaccharides and polypeptides as the three main types of macromolecules. Let me be clear about what goes into a nucleotide. There are three separate parts. First, you have the heterocyclic compound AHES. That's the nitrogenous base we talked about. Second, you have a monosaccharide AHE, a sugar. Third, you have a phosphoric acid or phosphate group. These three components linked together make a complete nucleotide. The five bases are adenine, guanine, cytosine, uracil, and thymine. You know them by their letters, A, G, C, U, and T. All of them are heterocyclic compounds, S-nephramkurchin olefins. They have ring structures with nitrogen atoms in them. Now, we categorize these bases. Adenine and guanine are called purines. They have two rings in their structure. The other three, uracil, cytosine, and thymine are called pyrimidanes. They have one ring. This structural difference is important for how DNA and RNA pair their bases. Here's the critical difference between DNA and RNA. The sugar component can be one of two types. Ribose is a 5-carbon sugar, a pentose. 2'-deoxyribose is also a 5-carbon sugar, but it's missing an oxygen atom compared to ribose. That small difference makes a big difference. When nucleotides are linked together with deoxyribose as the sugar, you get DNA, deoxyribonucleic acid. When they're linked with ribose as the sugar, you get RNA, ribonucleic acid. That sugar difference is actually how they got their names. Both DNA and RNA function as genetic material, storing and transmitting hereditary information. Now we're going to explore protein structure in detail. But first, let me point out something interesting. When scientists talk about structure, they mean different things depending on their field. Inorganic chemists write molecular formulae, semodicity. That's their structure. Organic chemists draw two-dimensional pictures. Physicists visualize three-dimensional arrangements. Biologists, though, we have our own approach. We describe protein structure at 4 different levels. 4. Each level adds more complexity and more information. Each level is important because proteins need all 4 levels of organization to do their jobs properly. Primary structure is the simplest level. It's the sequence of amino acids. Which amino acid comes first? Which is second, third, and so on down the chain. This sequence, this order, sequence, is critical. Change the sequence, and you change the protein completely. We represent protein sequences as a line. The left end represents the first amino acid that was made, polypeptides. We call that the N-terminal amino acid. The right end represents the last amino acid, peptides, the C-terminal amino acid. This direction is always written the same way, leb-to-les. N to C, left to right. It's a convention that helps us communicate clearly. Now, a protein doesn't just exist as a straight line. It folds. We call local folding patterns secondary structure. The most common secondary structure is a helix. Imagine a spring or a revolving staircase. That's actually a great description because when you see a helix from the side, it kind of looks like a staircase spiraling up. Here's an interesting detail. All the helices in proteins are right-handed. Not left-handed, always right-handed. That's just how proteins are. And not every part of the protein forms a helix. Some regions do, but other regions fold into different shapes. There might be extended regions, or loops, or other arrangements. Beyond helices, there are other secondary structures. Different parts of the protein can fold differently. What we call secondary structure is really the sum of all these local folding patterns throughout the protein. These structures are held in place by hydrogen bonds over specific, specifically bonds between atoms in the backbone of the protein chain. Now we step up to tertiary structure. This is the three-dimensional shape of the entire protein. Imagine taking that protein chain with all its secondary structures, all those helices and loops, and folding it further upon itself. It gets compacted, crumpled, folded into a specific 3D shape. It's kind of like a hollow woolen ball, lots of folds and crevices. This tertiary structure, this 3D shape, is absolutely critical. It's not just decorative, the shape determines where pockets and crevices are. Those pockets are where other molecules can bind. That 3D structure is what makes a protein function. Without the right tertiary structure, a protein is useless, even if it has the right amino acid sequence. The shape is essential. Finally, there's quaternary structure. Here's where it gets interesting. Some proteins aren't just a single chain. They're made of multiple protein chains, abbreviable polypeptides, all assembled together. Each chain is called a subunit. Quaternary structure describes how those subunits arrange relative to each other. The science has been clear for a long time, but the evidence from this past decade is definitive. This period from 2015 to 2025 will be remembered as the decade the consequences of our inaction became undeniable. That temporary breach of 1.5 degrees served as a final, unambiguous warning. Specifically, being a hemoglobin has 2 alpha-type subunits and 2 beta-type subunits. We write it as 2 alpha and 2 beta. The way these 4 subunits assemble together their quaternary structure is what makes hemoglobin work as an oxygen carrier. Change that quaternary structure and hemoglobin can't do its job. Let's talk about one of the most important jobs proteins do here, acting as enzymes. Enzymes are biological catalysts. Almost all enzymes are proteins. But here's an interesting exception. Some nucleic acids, specifically RNA, can also act as enzymes. We call these ribozymes. They're rare, but they're important. Enzymes can be drawn as simple line diagrams, and like any protein, they have all 4 levels of structure: primary, secondary, tertiary, even sometimes quaternary if they have multiple subunits. But when you look at the 3-dimensional structure, something special happens. The chain crisscrosses itself. It folds back on itself. This creates crevices and pockets throughout the 3D structure. Now one of those pockets is special the substrate, the molecule the enzyme works on, fits into this active site like a key in a lock. That active site shape is very specific. Different enzymes have different active site shapes. That's why each enzyme recognizes and works with just one specific substrate, or maybe just a few related substrates. We call this substrate specificity. Through that active site, enzymes catalyze their reactions at incredibly high rates. Here's an important difference between enzymes and the inorganic catalysts you might use in a chemistry lab. Those industrial catalysts, they work great under extreme conditions. High temperatures, high pressures. That's how they speed up reactions in manufacturing. But enzymes, they're fragile. They're proteins, and proteins are delicate. Above about 40 degrees Celsius hemoglobins with just slightly above normal body temperature cephalon. Enzymes start getting damaged. The heat causes them to denature. That means they unfold, their 3D structure collapses, and they stop working. Usually this damage is permanent. But there's an interesting exception. Some organisms live in extremely hot environments, hot vents on the ocean floor, sulfur springs. The enzymes from these thermophilic organisms are different. They're stable even at 80 to 90 degrees Celsius. Thermal stability is a key characteristic that evolution selected for in these organisms. Scientists and biotechnology companies love these enzymes because they're so hardy. Before we dive into enzyme mechanisms, we need to understand what a chemical reaction actually is. There are 2 types of changes that matter to us. First is a physical change. That's when something changes shape or appearance, but no bonds are broken. It's still fundamentally the same substance. State changes are physical processes. When ice melts into liquid water, that's physical, you still have H2O, just in a different state. When water evaporates into steam, again, still H2O, just as a gas. No bonds breaking, no new substances forming. A chemical reaction is different. In a chemical reaction, bonds break, new bonds form, atoms rearrange. You get new substances with different properties. For example, if you combine barium hydroxide with sulfuric acid, you don't just change the state, you create barium sulfate in water. Those are completely different compounds from what you started with. That's a chemical reaction. En is mekaniskt distinction etheria ris tipi en identification warhead. Navi, dufani, thershin leak good. Of the guessing formas with medium bottomala in chupifik that we in common state can you can see a gift both gas is up with pair expert beef and you actually I don't know where should I and let it stay even Now reactions happen at different speeds. We measure this with reaction rate now, how much product gets made per unit of time. If a lot of product forms quickly, that's a fast reaction. If you have to wait a long time for a little product, that's slow. Mathematically, we express rate as change in product divided by change in time. When you specify direction, we sometimes call rate the velocity of the reaction. Now, what affects rate? Temperature is huge. There's a general rule of thumb makes for every 10-degree change in temperature, the reaction rate approximately doubles or halves. Increase temperature by 10 degrees, rate roughly doubles. Decrease by 10 degrees, rate drops by about half. Here's where catalysts come in. When a catalyst is present, any catalyst, the reaction rate increases dramatically. It goes way faster than it would naturally. With enzyme catalysis, the rate increase is even more dramatic. Enzymes are incredibly efficient catalysts. Let me give you a concrete example of enzyme power. Consider this reaction. Carbon dioxide plus water produces carbonic acid. This reaction is happening in your blood right now. It's crucial for respiration and maintaining pH balance. But without a catalyst, this reaction is incredibly slow. If you just mix carbon dioxide and water and waited, how many carbonic acid molecules would form in an hour? About 200. That's it. For such a critical reaction, that's impossibly slow. Your body couldn't survive with such a slow rate. Now, your cells have an enzyme called carbonic anhydrase. When this enzyme is present, the rate skyrockets to about 600,000 molecules of carbonic acid formed every single second. Not per hour per second. That's an acceleration of roughly 10 million times. The power of enzymes is absolutely incredible. That's the difference between life and death. Reactions don't usually happen in isolation. They're connected. A series of reactions where the product of one reaction becomes the starting material for the next to get. We call that a metabolic pathway. Each step is catalyzed by an enzyme, maybe the same enzyme if it's versatile, or usually different enzymes for different steps. Here's a key example: glucose being converted to pyruvic acid. Glucose starts the pathway. It goes through 10 different enzyme catalyzed steps. At each step, a different enzyme acts on the product of the previous step. By the end, you have pyruvic acid instead of glucose. This pathway is central to cellular respiration depths, how your cells get energy. Here's something remarkable about metabolism. The same pathway, the same initial steps, can lead to different products depending on conditions. That's metabolic flexibility, the ability to adapt to different situations. Let me show you examples. In your skeletal muscles during intense exercise when oxygen is limited and aerobic conditions, the glucose pathway produces lactic acid. That's why your muscles burn during hard exercise. Under normal aerobic conditions in muscles, the same pathway produces pyruvic acid, which can be used for energy. But in yeast cells during fermentation, that same pathway produces ethanol, alcohol. Same starting material and first steps, completely different end product. Now, let's understand how enzymes actually work at the molecular level. First, understand what a substrate is. It's the molecule the enzyme is going to convert into a product. For this to happen, the substrate has to get to the enzyme and bind there. The science has been clear for a long time, but the evidence from this past decade is definitive. This period from 2015 to 2025 will be remembered as the decade the consequences of our inaction became undeniable. That temporary breach of 1.5 degrees served as a final, unambiguous warning. The science has been clear for a long time, but the evidence from this past decade is definitive. This period from 2015 to 2025 will be remembered as the decade the consequences of our inaction became undeniable. That temporary breach of 1.5 degrees served as a final, unambiguous warning. When the substrate is bound to the enzyme inside that active site, the substrate doesn't just sit there. Its structure changes. It forms what we call a transition state structure. This is a special, high-energy form of the substrate, kind of like the maximum distortion the molecule goes through. The enzyme then breaks and forms bonds. The substrate structure transforms into the product structure. The pathway from substrate to product must go through that transition state. There might be other intermediate structures along the way, but these are all unstable mutuous. They don't last. They're just stepping stones to the final product. Why are these intermediate states unstable? Stability depends on energy. Higher energy means less stable. These intermediate forms have high energy, so they're unstable. Only the substrate and product have relatively low energy and are stable. Everything in between is high energy and unstable. Scientists visualize this with energy diagrams. On the y-axis, you plot potential energy. On the x-axis, you show the progression of the reaction, the substrate transforming into product. The diagram shows the energy level of the molecule as it transforms. On that energy diagram, compare where the product sits versus where the substrate sits. If the product is at a lower energy level, that's an exothermic reaction. It releases energy. If the product is higher, that's endothermic molus. It requires energy input. With exothermic reactions, you don't need to heat anything. Once it starts, it proceeds because the product is lower energy than the substrate. With endothermic reactions, you have to keep supplying energy to drive the reaction. But here's the crucial point. Whether the reaction is exothermic or endothermic, there's something they have in common. The substrate has to go through a transition state with much higher energy. The substrate has to climb that energy hill. The difference in energy between the substrate and the transition state, that's what we call the activation energy. Activation energy is absolutely central to understanding enzymes. It's that energy hurdle the substrate has to overcome. Reactions with high activation energy are slow peptides. The substrate rarely has enough energy to make it over the hill. Reactions with low activation energy are fast substrates can easily get over. This is where enzymes work their magic. Enzymes bring down that energy barrier. How do enzymes do this? The enzyme stabilizes the transition state. That peak of the energy hill gets lowered. The transition state doesn't require as much energy anymore, so the substrate can get from S to P much more easily. The energy barrier is reduced. That's how enzymes speed up reactions so dramatically become incompatible because in his markle from Biomolecules. Let me review the actual mechanism of enzyme action. Enzymes have a substrate binding site, that's the active site we discussed. When substrate binds there, a highly reactive enzyme substrate complex forms. This ES complex is very reactive, ready to transform. But it's short-lived. Almost immediately it falls apart into product and the enzyme, which is unchanged. Actually, there's an intermediate step. Between the ES complex falling apart and the product being released, there's a brief enzyme product complex, EP. It's part of the complete cycle. The full cycle is enzyme plus substrate reversibly form the ES complex, which goes to the EP complex, which then releases product and regenerates the free enzyme. The ES complex formation is absolutely essential. Without it, nothing happens. That's why substrate specificity is so important. The substrate has to fit the active site and form that complex. Only then can catalysis occur. Let's walk through the 4 steps of the catalytic cycle. Step 1 is binding. The substrate finds the active site and binds there. Remember, shape specificity matters. The substrate fits into the active site. Step 2 is the induced fit. When the substrate binds, the enzyme doesn't just passively hold it. The enzyme changes shape. It wraps more tightly around the substrate. This movement is important to piezoelectric. It positions catalytic residues perfectly for the next step. Step 3 is the catalytic step, the transformation. The active site, now perfectly positioned around the substrate, breaks the substrate's chemical bonds. The enzyme facilitates this bond breaking. As old bonds break, new bonds form, and the enzyme product complex is created. Step 4 is product release and renaturation. The products are released from the enzyme. The enzyme returns to its original shape. Now it's ready to do it all over again. It can bind to another substrate molecule and run through the cycle again. This is why enzymes are so efficient. They can repeat this cycle thousands of times per second. Many things can affect how well an enzyme works. Any condition that changes the enzyme's tertiary structure, its 3D shape, can affect activity. A small change in shape can destroy the active site's function. Let's look at the main factors that affect enzyme activity. Enzymes don't work well everywhere. They work in narrow ranges of temperature and pH. Too cold, too hot, too acidic, too basic enzymes, and the enzyme's activity drops. Each enzyme has its own optimum temperature and optimum pH. For example, human enzymes typically have an optimum temperature around 37 degrees Celsius normal body temperature. At that temperature, they work best. Below or above that, activity drops. Same with pH. Each enzyme has a pH where it works best. Below or above that pH, activity decreases. Temperature effects are interesting. Low temperature preserves the enzyme in a temporarily inactive state. That's why labs refrigerate samples and why you refrigerate medicines. The enzyme isn't destroyed, it's just inactive. You warm it up and it works again. But high temperature is different. Heat denatures the protein. The 3D structure collapses. The active site is destroyed. This damage is usually permanent. The enzyme is gone. Now let's look at substrate concentration. As you add more substrate, the reaction goes faster. More substrate means more chances for enzyme-substrate complexes to form. More complexes forming means more product being made per unit time. So velocity increases. But there is a limit. Eventually, the reaction reaches a maximum velocity, microls, we call it Vmax. After that, adding more substrate doesn't make the reaction faster. The curve plateaus. Why? Because the enzyme molecules are saturated. Here's the reason. There are only so many enzyme molecules present, but there can be way more substrate molecules. After a certain point, every single enzyme molecule is working on a substrate. They're all busy. There's no free enzyme sitting around waiting for more substrate. All the existing enzymes are running at maximum speed. That's Vmax. You can't go faster until you have more enzyme. Enzymes can also be controlled through inhibition. Certain chemicals can bind to enzymes. When they do, the enzyme's activity decreases or stops. We call these chemicals inhibitors, and the process is called inhibition. One important type is competitive inhibition. The inhibitor molecule looks like the substrate. It has similar molecular structure. Because it looks similar, it can also fit into the active site. Now here's the competition. Both the inhibitor and the substrate are trying to bind to the same active site. Sometimes the substrate gets there first, sometimes the inhibitor does. If the inhibitor binds, nothing happens to ultimately. It's not transformed into a product. The enzyme just sits there with the inhibitor attached. As inhibitor concentration increases, inhibitors win more of the competitions, and enzyme activity drops. The classic example is malonate inhibiting the enzyme succinic dehydrogenase. Malonate closely resembles succinate, which is the real substrate. Malonate looks like succinate so well that it can bind to the enzyme. But once bound, nothing happens. Malonate just sits there, blocking the real substrate from binding. This type of competitive inhibition is actually used medically to control bacteria allergic to Carpaudia. It blocks bacterial enzymes without harming human enzymes that work differently. Scientists have discovered thousands and thousands of enzymes. Each one was isolated, studied, and classified. The classification system is based on the type of chemical reaction each enzyme catalyzes. There are 6 main classes. Each of these 6 classes has multiple subclasses between 4 and 13 for each class. Each specific enzyme gets assigned a 4-digit classification number that tells you which class, which subclass, and exactly what reaction it catalyzes. Let's look at the 6 classes. Oxidororeductases, also called dehydrogenases, are the first class. They catalyze oxidoreduction reactions, matrix terms. They transfer electrons from one molecule to another. One molecule gets oxidized, the other gets reduced. Transferases are the second class. They transfer groups between molecules. The transfer group could be a methyl group, a phosphate group, an amino group, various groups, but specifically not hydrogen. If they transferred hydrogen, they'd be in the oxidoreductase class. Hydrolyses are the third class. They break bonds, and they do it using water molecules. That's why it's called hydrolysis. They can break all sorts of different bonds, ester bonds in fats, peptide bonds in proteins, glycosidic bonds in polysaccharides, and others. It's a big, diverse class. Lyases are the fourth class. They remove groups from substrates, but they don't use water to do it. They use different mechanisms. When they remove a group, they often leave a double bond behind. Decarboxylases remove carboxyl groups. Dehydratases remove water, creating double bonds. Isomerases are the fifth class. They don't break or add bonds between molecules. Instead, they rearrange atoms within a single molecule. They convert different isomers into each other molecules with the same molecular formula, but different arrangements of atoms. Finally, glucases are the sixth class. They do the opposite of hydrolases. They join 2 compounds together. They form new bonds in our carbon-oxygen bonds, carbon-nitrogen bonds, phosphorus-oxygen bonds, and others. Glucanases usually need energy to work, often from ATP. They're sometimes called synthetases. DNA ligase is a famous example. It joins DNA fragments together. Many enzymes are just proteins, but many others need more than that. They need non-protein components. These components are called cofactors. A cofactor is any non-protein substance that an enzyme needs to be catalytically active. When you have just the protein part of an enzyme, it's called an apoenzyme. By itself, an apoenzyme is inactive mechanism. It can't work. You need the cofactor. When the cofactor binds to the apoenzyme, you get an active enzyme. The complete active enzyme. That complete active enzyme has a different name. We sometimes call it a holoenzyme. There are 3 types of cofactors. First, prosthetic groups. Second, coenzymes. Third, metal ions. Each type has different characteristics. Prosthetic groups are organic compounds, carbon-based molecules. The key characteristic is that they're tightly bound to the protein. They don't just float around loosely, they're securely attached. Perfect example, the enzymes peroxidase and catalase both break down hydrogen peroxide into water and oxygen. Both use heme as their prosthetic group. Heme is actually part of the active site. It's crucial for catalyzing the reaction. The enzyme doesn't work without its heme. Coenzymes are different. They're also organic compounds, but they're not tightly bound. Their association is transient, temporary. They bind to the enzyme during catalysis, help with the reaction, then separate. Here's something interesting. Coenzymes can work with many different enzymes. One coenzyme can participate in reactions catalyzed by several different enzymes. They're versatile. They actually participate in the reaction. They're like co-substrates. Now here's crucial information. Many coenzymes have vitamins as essential components. NAD, nicotinamide adenine dinucleotide, money contains niacin, a B vitamin. NADP also contains niacin. This is exactly why vitamins are essential in your diet. Without them, your enzymes can't function. You can't make these coenzymes. Your metabolism falls apart. Some enzymes need metal ions to work. Zinc, iron, magnesium, Fe, various metals. These metal ions form coordination bonds with the enzyme's amino acids at the active site. They also interact with the substrate. This positioning is crucial for catalysis to happen. A great example is carboxypeptidase. It's an enzyme that breaks peptide bonds. Proteases do this. Carboxypeptidase requires zinc as a cofactor. The zinc is essential. Without it, the enzyme is useless. Here's the bottom line on cofactors. If you remove the cofactor from an enzyme, catalytic activity drops to zero. The enzyme stops working. The Apple enzyme alone cannot function. This proves how crucial cofactors are. They're not optional extras. They're absolutely required for the enzyme to work. Let's wrap up by stepping back to see the big picture. We looked at everything from small molecules to complex enzymes. Here's what's remarkable. Despite the incredible diversity of life on Earth, all those different organisms, their basic chemistry is remarkably similar. The same metabolic reactions happen in bacteria, plants, animals. The fundamental chemistry of life is constant across life. When you analyze the elements chemically, living tissue and non-living matter contain similar elements, but the proportions are different. Carbon, hydrogen, and oxygen are much more abundant in living organisms. That difference, that higher proportion of C, H, and O means less, that's what distinguishes life from non-life. And the most abundant chemical, water. Every single living organism is mostly water. Everything else, all the proteins, the lipids, the nucleic acids, all the reactions, they all happen in water. We discussed thousands of small biomolecules, molecules with molecular weights under 1,000 Daltons. Amino acids, sugars, fatty acids, nucleotides, nucleosides, and nitrogen bases. Remember the key numbers. 20 types of amino acids in proteins. 5 types of nucleotides. These numbers are universal, the same across all organisms. These are the universal building blocks of life. Fats and oils are glycerides, fatty acids attached to glycerol. Phospholipids are fats with added phosphate-containing compounds. They are crucial for membranes and energy storage. For macromolecules, only 3 types: proteins, nucleic acids, and polysaccharides. They're all polymers made of repeating units, just different building blocks. Macromolecules have that hierarchy of structure we discussed. 4 levels for proteins especially. Each level is important for function. Nucleic acids carry hereditary information. They pass that information from parents to offspring. This information is encoded in DNA and RNA. Polysaccharides have structural roles of cellulose in plant cell walls, chitin in arthropod exoskeletons. They also store energy starch and glycogen. Proteins are incredibly diverse. They're enzymes catalyzing reactions, antibodies fighting infection, receptors receiving signals, hormones coordinating activity, structural proteins providing support. Collagen is the most abundant protein in animals. Rubus yo is the most abundant protein on the entire planet. Enzymes catalyze biochemical reactions. Almost all are proteins. Ribozymes are the exception, mRNA, that can catalyze reactions. Enzymes show substrate specificity. Each enzyme recognizes its specific substrate. They need optimum temperature and pH. They're damaged by high heat, permanent damage. Most importantly, enzymes lower activation energy. They dramatically increase reaction rates. They make reactions fast enough to actually sustain life. Without enzymes, life would be impossibly slow.
Meet Iyal, your AI study companion. Transform PDFs into smart study guides, practice tests, and flashcards for UPSC, NEET, banking, and any competitive exam.
Master your exam preparation with Iyal.